Pleasure to be here and to talk to your audience. I work in a very esoteric field, and I realize that you kind of have to have many years of schooling, of education, to kind of understand the terminology. So it's my job today is to try and make it more accessible, so you understand what I'm trying to do and how this is helping patients with Parkinson's disease. And I thought I'd start off by telling you a little bit about where I'm from. I'm English originally, and the picture on the screen in front of you is basically Devon. It's Dartmoor. It's and it's an asylum on Dartmoor called Moorhaven, where when I was young I used to spend my summers. It's at the time, it's now luxury apartments. At the time it was basically a residential care facility for people with neurologic diseases, all sorts of neurologic diseases and psychiatric disorders. And when I was a child, I realized that from a very early age that there has to be a better way of understanding problems of the brain, diseases of the brain, and there has to be a better way of trying to help patients with disorders in neurologic function. And so that kind of set me on a career trajectory to try and understand all sorts of different brain disorders. I worked in children's homes subsequently with kids with Down syndrome and cerebral palsy. And then I went on to work in cognition and Parkinson's disease subsequently. But they're all neurologic disorders. And one of the things I realized very early on is a great way to access the brain and to try and understand what's going on in the brain at the most fundamental level is through genetics. So that's where I started. I did a PhD in statistical and molecular genetics and what's known as complex trait mapping, which basically means trying to find out what are the genetic influences that underlie seemingly sporadic diseases. So this is where I am now. This building, it's we're on the 5th floor. It's a lovely purpose-built building, and it's got 2 floors of neurology clinics and 3 floors above of kind of wet neuroscience labs. And I'm going to tell you a bit about genetics of Parkinson's disease. So I'll start with this slide. It's about Parkinsonism and related disorders. Now, when I started in the in my area, which is the molecular basis of Parkinson's or the genetics of Parkinson's, individuals that had a family history of Parkinson's disease were considered not to have Parkinson's disease, but to have Parkinsonism. They had to be referred to as by symptoms rather than by the disease definition. And the reason for this is that classically, Parkinson's disease is defined as, okay, this constellation of movement problems, but also by pathology, end-stage pathology in the brain when a person dies. And that typically consists of these inclusions, these protein inclusions within cells and cell loss in the substantia nigra. And you can see on this picture, you can see here, I don't know if you can see my mouse. Can you see my mouse, Caitlin? Maybe not. But there's a substantia nigra is labeled there. It's in the brainstem, top of the brainstem, in the midbrain here, just above the brainstem. You can see it here. And this structure, basically this part of the brain, is responsible for producing dopamine that innervates the striatum just above the putamen and caudate nucleus here. There are many forms of Parkinsonism, though. Parkinson's disease is the one we know best and is recognized most, and it's the most common, of course. But there's also other forms of neurologic complaints with Parkinsonism as a feature that have lots of Lewy bodies. The next one down is, for example, dementia with Lewy bodies. That's also a relatively common complaint. It tends to fall in the cracks between memory and movement disorders in clinics. There's many other disorders here, multiple system atrophy, and then becoming more and more rare, for example, neurodegeneration with brain iron accumulation. But there's also other forms of Parkinsonism known as tauopathies, and types of Parkinsonism when the patients don't have either Lewy bodies, alpha-synucleinopathy, or tauopathy, and have other types of pathologies, but nevertheless develop symptoms of Parkinsonism as well. So really, Parkinson's disease per se, to me, is what I call an umbrella term. It's basically, there's lots of things that come under that title, if you like, under that definition, and they include all of these different disorders, really. So what does my group do? Well, the mission really is to predict and prevent neurologic diseases, and our major focus is Parkinson's disease and has been for more than 20 years. So we start here on the top kind of right-hand side of the diagram here of this circle, in what I call neurology and neuropathology. Here's a picture, by the way, of a midbrain that's been cut through, and you can see, let's move this from my screen. You can see in the top image is a cut through. It's kind of a, what do you call it? Oh, things are moving, not by my accord. Oh, yeah. Sorry. Yeah, there's a picture of the substantia nigra. Substantia nigra means substance black. It's a black substance in the brain. It's the dopaminergic neurons that innervate the striatum. And in the top part of the image, you can see a normal substantia nigra from someone who's deceased and given their brain to autopsy. And in the bottom picture, you can see a substantia nigra that's a little bit burnt out. It's not so pigmented. It's not so black. And that probably came from a patient with Parkinson's disease. We start here on the right-hand side of the diagram, thinking about population and family-based genetics. And then we go through this circle, this virtuous circle, I like to think of it, through using that information that we find in those families to develop mouse models of the condition. It's really mouse models of gene dysfunction that we've identified within the families, and we use those models basically to start thinking about how to prevent that gene dysfunction and ultimately prevent the symptoms that you might find in those families with those particular forms of Parkinsonism. So you go round to the left-hand side of the diagram. And part of this leads to new opportunities for imaging in human beings as well as in mice. And here's a picture of a rat, actually, the imaging done in a rat model alongside imaging done in a human patient using 18-fluorodopa. So I'm going to tell you a bit about it, and it's going to be a bit of a historical overview and a bit of a review, and I'll look forward to the questions at the end. So it starts here with families. This is the first family that I worked on with Parkinson's disease. It's known as the Spellman-Munter kindred, and I met Manfred Munter back in 1997. He was the chair of neurology at Mayo Scottsdale, and I was at Mayo Clinic at the time. And he presented to me a paper that he'd been working on. He called it his life's work, and it was basically the biochemistry of the brain of 4 members of this particular family. Now, the lady here in the middle, holding the child in her arm, this photograph's from 1924. The lady here in the middle of the photograph, you might notice her arm's a little bit contorted. It's a little bit strange, her posture. It looks a little dystonic. And you might notice her face. Her face looks a little masked. She's staring on the photograph, and rather than smiling, she's got an open mouth, as you can see, and she's just staring into the camera. And of course, back in the day when these types of photographs were taken, you had to hold the pose for a very long time to to get the picture. This lady has a form of early onset Parkinsonism, and so does her other family members. The average age of onset in the family was 33, and the average duration of death from the disease, from this form of Parkinson's disease, was 10 years. Well, I met Manfred and I talked to him about the family, and he introduced me to 1 or 2 of the living family members. Um, and although Parkinson's disease at the time was not considered to have any genetic basis, he basically encouraged me to look into this. And as a geneticist, you know, it was a bit like a red rag to a bull, really. I decided to take it on. This is very much against the view in the field at the time. There was no genes for Parkinson's disease back then when I started this, and people considered it a rather futile and silly expedition to try and understand the disease through its genetic basis. Fast forward to today, and I can tell you there's about 200, well, there's more than that. There's probably about 340 genes now that have been implicated in Parkinson's disease. So it's actually a very rich, fertile ground to find genetic, to understand the disease through its genetic basis. Anyway, through Manfred's introduction, I was very privileged to meet many different family members, and I soon learned that they'd all run away. They've all tried to escape the bug. They'd been told that living on the farm where they were, that there was something in the environment, something on the land that was causing their disease. And so they'd scattered. All the different family branches have scattered across the United States to different corners and set up new homes. But of course, the bug followed them. And it was quite tragic to visit many family members and to hear their stories. But they were incredibly generous. And it made me incredibly humble because they trusted me with some DNA samples, some blood samples, and I told them that we'd find the gene and we'd do something about it. And sure enough, we did. So the next slide's a bit of science. And on the top of the slide is pedigrees. Some of you might be familiar with looking at these types of things. I'm not sure, but basically what you see here, for example, this top one. There's a 3-generational family. There's grandparents at the top. Squares are males, circles are females. And as you go down, you can see the next generation of siblings and the following generations of siblings. And so basically, it's a picture, if you like, of family relationships. The people denoted with black symbols are people who have Parkinson's disease. And these are examples of families with synuclein multiplications. After about 5 years of effort, of tracking down every family member from the Spellman-Munter kindred, we eventually identified what the molecular cause was. And it turned out to be a region of chromosome 4, 4q21, that was multiplied. Whereas you and I have 2 copies of this region of chromosome 4, people with the disease in the Spellman-Munter kindred and in many other families now actually have 3 copies or even 4 copies. In the Spellman-Munter kindred, they had 4 copies. On the left-hand side of the screen here, at the top, this blue circle, it's not a blue moon, it's basically staining of DNA in the nucleus of a blood cell. And what we've done is labeled up 2 probes for the gene, alpha-synuclein. For a 5' piece of the gene, we've labeled up with a red probe, and the 3' end of the gene we've labeled up with a green probe. And when they coalesce together, you can see there's kind of a yellow signal that emerges, or you can see the 2 signals together. So you can see in a normal nucleus, there's 2 copies. But in some folks with Parkinson's disease and this particular molecular cause, they have maybe 3 copies. You can see in the middle section there. And in the lower left quadrant, you can see that there's a nucleus there of a blood cell labeled in blue, and you can actually count. There's 4 distinct copies of the alpha-synuclein gene. And what this told us was something quite profound. If you look in the right-hand side, bottom of the slide there, you can see that people who have 3 copies, 3 copies of this particular gene, you can predict at what age they were likely to manifest with disease symptoms. And typically by around 50 years of age, for example, 50% of people with that particular genetic makeup will be affected with Parkinsonism. On the other hand, people who have 4 copies of the alpha-synuclein gene, 3 on their mutated allele, by the age of 50, probably something in the order of 80 to 90% of those individuals will already be affected with Parkinson's disease. And it turns out that the dose, the amount of alpha-synuclein being expressed, is directly related to the age of onset of Parkinson's disease in these families. And that tells us something very important, because if that's the case, then maybe if we lower the amount of alpha-synuclein being expressed, we have a therapy, we have a way forward to prevent these individuals getting the disease. And of course, we have this molecular assay to be able to predict it in the first place. It turns out, so back in 1998, we first developed some antibodies. These are reagents basically that can be used to identify the protein that's encoded by the synuclein gene. It's known as alpha-synuclein as well. And we made some antibodies, and these antibodies basically, these are some slides, some sections of brain tissue, A, B, C, D. These are very thin sections of brain tissue from patients with Parkinson's disease. And all of the brown stuff is basically alpha-synuclein pathology. And I remember at the time Dennis Dixon came running down the hallway. He's a neuropathologist at Mayo. And he was so, well, excited, and perhaps that's the wrong term to use, but he was profoundly excited. He had never seen anything like it. The whole brain of these folks that he had in his brain bank was lit up with synuclein pathology. It's occult pathology that no one really had seen before in this type of detail. And you can see these like thread-like structures. These are Lewy neurites. These are basically processes that are emanating from neurons, and they're chock-a-block full of alpha-synuclein. You can also see very classic Lewy bodies. There's in section A in the top left, you can see this round-like circle. Looks like a moon-like structure, with this very densely filled, densely stained halo of alpha-synuclein. That's a classical Lewy body. And we've learned since that basically this pathology, and using these types of reagents, alpha-synuclein antibodies, that the synucleinopathy in Parkinson's disease in many patients with Parkinson's disease seems to come from 2 places originally. It seems to manifest first, strangely enough, in the brain stem, and perhaps even in the gut, certainly through the vagus and via the vagal nerve, into the brain stem, and then ascends very gradually over the years into the midbrain, which is the part of the brain that that basically produces dopamine and innervates the striatum, which is also shown in the middle. You also get synuclein coming, this Lewy body pathology, you see it first in the olfactory bulb too. So it seems to come in somehow. Well, it seems to, I don't know whether it's invasive or what, we don't know. The pathology appears to manifest in the olfactory bulb and into the brainstem, and then ascends through to the midbrain, and then ultimately in late onset, typical garden variety Parkinson's disease, it may manifest into the cortex. So this is this kind of ascending pathology. Now, in these families that I've been telling you about, alpha-synuclein multiplication, overexpression of synuclein, is the cause of their disease. And you know, this is they have a lot of this type of pathology in the brain. They have a lot of movement problems, and they ultimately have a lot of cognitive problems too. In most patients with Parkinson's disease, there isn't an alpha-synuclein multiplication, and but nevertheless, the pathology, the synuclein pathology, seems to follow the same pattern. So we've looked genetically at alpha-synuclein in people with garden variety Parkinson's disease, not just the rare families with inherited forms. And what we find is something quite peculiar. On the top here, on the top, you see a little graph, and it's labeled from 1 to 21. 22 is not shown, but it is shown. The number isn't 22 is not shown, but 1 to 21. These are the chromosomes. So chromosome 1, 2, 3, 4 in the human genome. And the signals shown above are basically regions of the human genome where there seems to be a difference between individuals with Parkinson's disease and individuals who are otherwise healthy and are matched in terms of their background ethnicity. And you can see here on chromosome 4, actually it's chromosome 4q1, there's SNCA, it's labeled here, that's the symbol for alpha-synuclein. It turns out that alpha-synuclein is the largest hit, and this is garden variety Parkinson's disease. So there's basically there's a very strong signal, but it's indicating very modest risk in many patients with Parkinson's disease in and around the alpha-synuclein gene. And we've dissected that using a technique known as high throughput sequencing to try and understand where that signal is coming from. Now, in some folks, the difference, and it's a very modest difference in patients with Parkinson's disease versus otherwise healthy people. And so in some folks, the difference basically can be attributed to leading to the movement disorder, the Parkinsonism, and that's happening towards the 3' end of that gene. And in other folks, when they had Parkinson's disease, but perhaps a more malignant form of Parkinson's disease, where there's also dementia that subsequently manifests and perhaps manifests early, there's also a signal at the 5' end of the gene. So you can even within the single piece of the genome, you can dissect it out to different influences, genetically speaking, that are contributing to two major features of Parkinson's disease. The good news is that those two pieces of information together, the idea from the families, it's more than an idea, it's a proven theory from the families that dose of alpha-synuclein, expression level of alpha-synuclein is directly correlated with age of onset and severity of the condition. That piece of information, along with the idea that garden variety Parkinson's disease in populations worldwide have something a little different. Those patients have something a little different, very modest risk in their alpha-synuclein gene as well, has basically spearheaded and encouraged many companies, mostly specialty pharma companies, to start thinking about therapeutics that might benefit patients with Parkinson's disease and might remove that synuclein pathology. And so many of these companies are making vaccines or specific antibodies or antibody fragments or other chemical-based methods to basically remove synuclein from the brain. And many of these compounds and drugs now, and I've got no bias here, I'm not involved in any of these companies' work, many of these companies now have got those medications in a variety of clinical trials in different parts of the world. So it's a really exciting time to see if this will work as a therapeutic. In those families with synuclein multiplication, I have no doubt that it will be successful. In patients with idiopathic garden variety Parkinson's disease, I'm not so sure. But it certainly will be of benefit, I think, for the potentially for the cognitive problems and potentially for people who have a more malignant, faster course of demise. I'm going to tell you a bit about what synuclein normally does. It's involved in neurotransmission, and it helps to regulate what's known as snare complex assembly, but I don't want to get too much in the weeds, but it helps to regulate the release, and in this little vesicle here, there's neurotransmitter, we can think of it as dopamine. It helps to regulate the release of that dopamine by helping that vesicle bind to the cell membrane and allow those contents, that neurotransmitter, to be released, and has a fundamental role to play in maintaining synapses, or the connections between neurons. I wanted to move on and tell you about another gene. So synuclein is a very big finding, and that really rocked the world and in many ways. And when I was, I started out in Parkinson's disease in early '97, and by 2003, the synuclein multiplications were published, and this news has basically started to break. 2004, we found the first synuclein duplication patients, and then we continued to find more and more. And I just published a paper with detailing 300 or so subjects with synuclein multiplications in 58 families. So more and more of these families are coming to light now. But that was thought still to be a rare thing. It's a very, it's interesting, the field thought, and sure enough, it might be interesting as a therapeutic, as a target. I think it's gained traction over the years. It certainly has. But nevertheless, people thought it was a one-off. But this next story I'm going to tell you about isn't a one-off, and it really took the genetics of Parkinson's disease to another level. It started off for me with working with these two families. One's known as Family D, or the Western Nebraska kindred, and one is Family A, a German-Canadian family, actually. It's part of the reason I moved to Vancouver, because of the work on this family. I had been working with these families for some years prior to a report that came out in the Japanese literature about another family known as the Sagamihara kindred, published by Fanyama Obata. And it showed that there's a region on chromosome 12 that the Sagamihara family seem to have inherited identical by descent in the patients in that family with Parkinson's disease. And the region was on a part of chromosome 12, 12q12. So we had a look in all of the families that we were working with. We looked in Family D and Family A. And sure enough, here's this little plot on the bottom right-hand corner. You can see there's, I don't know, it's statistical analysis basically of that region of the genome in these families. And the big peak there is basically evidence that there is indeed a region of chromosome 12.1.2 segregating down the family line from the founders of the family down through all the generations, basically. And this piece of chromosome is segregating with the disease. There's no logical analysis, linkage analysis. However, the big breakthrough came kind of for me in this with working with this fella here, Jan Asley. I met him back in about 1999 it was, or 2000, I forget, 99, around then. And I was saying to him at a meeting, actually it was in Japan, kind of funnily enough, and I was saying to him at a meeting in Japan that nobody in the field was very interested in what I was doing in the genetics of Parkinson's disease, because nobody really believed in it. And it was very hard for me to get my colleagues at Mayo Clinic to be interested in this, because they firmly believe that there was really very little evidence of a genetic basis for Parkinson's disease, and therefore it wasn't worth studying. And they wouldn't give me access to patients or to blood samples from patients. They just couldn't be bothered to do it, I suppose. And I said to Jan at the time, this is about in the late 90s, as I was sitting there crying in my coffee, kind of saying, I wish they would. And he said to me, Matt, you must be such a rich man. You have all these resources at Mayo. You have, they're wonderful, wonderful clinical records and epidemiology. And I said to him, no, I don't. And he said to me, well, I work on with many patients with Parkinson's disease, and I will ask them if they will be willing to share blood samples with you. And he was the chair of neurology in Trondheim at the time, in mid-central Norway, which is shown on the map there. And we started working together, and he shared with me many hundreds of patient samples. I got funding from the NIH to do the genetic work, and he got funding from the Norwegian state to do the clinical workup of the families. And in 2004, we identified a mutation called leucine-rich repeat kinase 2, G2019S, in many of these families. And these come from little fishing villages along the central and northern coast of Norway. It turns out that all these little families are shown here, and there's many more now, are actually all related back to the 15th century to one common founder, which is quite remarkable. And it turns out all the patients with Parkinson's disease in this family have a LRRK2 G2019S mutation. We realized that and what's shown in the bottom right-hand corner here, it's something quite unusual. Now, what's shown here in this table is as haplotypes. So it's basically, we're diploid organisms. We've got 2 copies of the human genome, each of us, one from our mum and one from our dad. And it's shown here that each of these individuals, their P numbers, proban numbers, represent one affected family member from each of the kindreds above. And what they have is each of these affected family members has a haplotype or a piece of chromosome inherited from one of their parents, which has the same genetic fingerprint as each other. So P052 has the same genetic fingerprint as P099 or P104 or P241. All of these individuals basically have the same piece of chromosome 12q12 inherited, identical by descent from their ancestor. They have the same genetic fingerprint. They're cousins, basically. They're cousins, at least for this part of the genome. They're very, very closely related. In fact, they're identical by descent. Over the right-hand side is what we find is something which is more surprising. I can understand the Norwegian families may be closely related because access from one village to another is by boat, and you can imagine that there would have been, romances over the years, and sure enough, the families would have been related. But on the right-hand side is also the same haplotype that's shared by families from other parts of the world, in the United States, in Ireland, and Poland. And we quickly realized that this wasn't just a few emigrants from Norway, but basically it's much older than that. It's I get the slides to move. It's much older than that, and it turns out that that basically almost everybody with a LRRK2 G2019S mutation is related to one ancestral founder. I'm going to tell you about that in a minute. LRRK2 at autopsy can lead to alpha-synuclein pathology, typical Lewy body pathology that I've shown you before. It can lead to, but it can also lead to other forms of pathology, like tau pathology and just unusual TDP43 or ubiquitin-based pathology in the brains of patients that clinically have exactly the same symptoms. They have clinically definite kind of Parkinson's disease. So that's very enigmatic. So enigmatic that actually it's Dennis who coined this term, that we consider now that LRRK2, the leucine-rich repeat kinase 2 gene, is basically the Rosetta Stone, if you like, of Parkinson's disease, of neurodegeneration in general, because it links together all of these pathologies that are found at end stage. And now we just have to understand what it's doing. We've got the beginning and we've got the end. We've got the gene and the mutations, and we've got the disease. We have to figure out the bit in the middle, the ontology, how this insult, genetic insult, predicates that clinical and pathologic picture. We found since, and this is shown in the top right-hand corner here, other variants. So this is a variant here which is very modestly increases risk. It's known as G2385R. It's also in LRRK2, leucine-rich repeat kinase 2. And again, it's related. People with the G2385R variant basically are all related together too. It actually is found in between 6 to 8% of all patients with Parkinson's disease in Asia, across Asia, all the way from Korea to Taiwan through mainland China and via Japan. So it's unfathomable to me, really, that the 6 to 8% of all patients with Parkinson's disease in Asia have a G2385R mutation, and that they've inherited it identical by descent from a common founder, and that the age of the mutation is around 4,800 years old. This is a paper that I published back in 2007. But with genetics, we can be very, very precise, and it's very difficult to argue that there's any other explanation. It's kind of black and white. We've also found that the LRRK2 variability in the gene LRRK2 can also be inversely associated with risk. So some people in the world without Parkinson's disease are not susceptible to to Parkinsonism because of their genetic variability in LRRK2. And there's a very interesting link here between LRRK2 and immunity. And so we might hear about in some webinars in the future, perhaps, is how the immune system and how the brain are talking to each other, and how LRRK variability is influencing basically protection to immune problems or immune compromise problems, but on the flip side, predisposing to Parkinson's disease in later life. So here's the G2380, here's the G2019S mutation, which is probably the most pathogenic and the most common mutation for Parkinson's disease in the world. It basically breaks the hinge of this purple ribbon here, the activation segment of the kinase itself within LRRK2, and keeps this kinase constitutively active within patients, just a little bit, 2 or 3-fold higher than in people without the mutation. And but of course, it leads us to think about, can we make a competitive LRRK2 specific kinase inhibitor? Because if we can block this site, we can perhaps restore LRRK2's normal functions, and with that, again, help to prevent Parkinson's disease. We can predict it based on genotyping individuals now, and perhaps we can prevent it. I went to the source of this in 2005, and I won't have time to tell you too much about it. I'm going to go pretty fast, but I was very fortunate to meet this fella here. His name is Faisal Hentati. I was talking to him last night, actually. He's the chair of neurology. He runs the National Institute of Neurology in Tunis. He's right down here in Tunisia on the north coast of Africa. It's where Carthage is, and you may have probably heard of Carthage. And Carthage was one of the city-states of the Phoenician Empire in around 3,000 years ago. And the reason I'm telling you this is because the Phoenicians are seafarers, and they had these trading states across the city-states across the Mediterranean basin, but they also did a lot of trading elsewhere, including around the coast of Spain, and including there's some contact, we think, with the with Norse peoples as well. The increased frequency of G2019S around the world is in part responsible to this type of trading, these types of population migrations, and we believe from genetic evidence that actually that LRRK2 G2019S mutation, that piece of chromosome, actually originates in and around Tunis and this part of the world in Berber peoples, and basically has spread. That piece of chromosome has spread around the world, so much so that most people with G2019S, like I say, are related to a common ancestor. In North Africa, in Berbers, 1 in 3 patients that comes into the clinic, 30% has a LRRK2 G2019S mutation, and so it's an enormous social healthcare burden in North Africa, and we're talking all the way from Morocco through to Libya. And we also find the same variant in Ashkenazi Jewish folks in Israel, and it's about 14% of patients that come into the clinic have the same LRRK2 G2019S mutation. So in this one, the mountain went to Mohammed, and I've been doing lots of work. I'm going to skip this one. This is just showing you the penetrance. Just because you have these genetic variants, by the way, if you do have a genetic variant like this, it doesn't necessarily mean that you're going to get Parkinson's disease. It basically just elevates the risk quite considerably with age, but it doesn't. It's not deterministic. It doesn't mean you're definitely going to get the condition. Parkinson's actually is what's known as a multifactorial disease. It's due to a combination of genetic background and exposure that you've had through a lifetime. So it's the two things together that's important. We've been doing a lot of work in Tunisia over the years, and basically looking with families, and this is a typical type of family from Tunisia. This is a recent study to try and find modifiers, genetic modifiers of age of onset in patients with LRRK2 G2019S. This is one that was published in 2016, known as variability in a gene called dynamin-3. We've been making mice models of LRRK2 G2019S, and this is showing some elegant work looking at the electrical properties of neurons in the brain of animals with a G2019S mutation. I'm not going to go into it today, but it appears that that these mice, here's a little figure. It appears that these mice basically have an increased probability of firing. These neurons in these mice, the candle's burning twice as bright, but perhaps half as long. They seem to have an accelerated form of synaptic senescence. The beautiful thing, though, of course, is that many drug companies now are sitting on LRRK2 kinase inhibitors. And some of those companies have been daring enough to go into clinical trials already with these, with the view that if they can apply competitive LRRK2 kinase inhibitors, it can indeed slow down or prevent the disease in people with these particular variants. And we're talking about a lot of people with Parkinson's disease in many different parts of the world. Whether those compounds have efficacy in garden variety Parkinson's disease, I don't know. But the companies involved are making a leap of faith based on reasonably good evidence that it will be of benefit, or certainly no harm to all patients with Parkinson's disease, to slow down or to halt the disease progression. So that's very exciting. It's just an illustration of having now a genetic predisposition to the disease allows us to find individuals who are asymptomatic, don't have any symptoms per se, not clinical symptoms manifesting, but allows us to engage them in research-based studies. And this is a study looking at the brain using neuroimaging and basically looking at dopamine release and serotonin release in the brain in individuals who are otherwise asymptomatic. They have no clinical symptoms of Parkinsonism, but nevertheless have, what we find is there's an increase in serotonin and in the brains of some of these individuals and in aggregate prior to any of them developing movement disorder. There's many other genes. I'm going to flip through them really fast because I've got to go and we're going to have some questions. There's Gaucher's disease. There's a mutation in many variants in GBA1 in glucocerebrosidase. Gaucher's is a lysosomal storage disease, but individuals who are carriers are heterozygous variants in GBA have been identified to have a particularly increased risk of a rather malignant form of Parkinsonism with a lot of alpha-synuclein pathology. Just point out this slide, just because you have a variant, for example, in GBA, doesn't necessarily mean you're going to get the disease. And here's an illustration of what I'm trying to say. For example, N370S is the most common variant in GBA. And in this little box here, you've got age 60, age 70, age 80 in the 3 different columns. And this is the increased risk of basically if you have an N370S, or indeed if you have any variant in GBA, what your increased risk is above people who don't. And you can see it's very modest. So non-carriers at the age of 80 have a 2.1% risk of Parkinson's disease, whereas people with GBA variants have a 7.7% increase of risk. It's very modest, and that's at the age of 80. It's a modest risk factor. And then nevertheless, it's very important because, again, it leads us to therapeutics. And several of these are now in clinical trial targeting individuals with GBA variants. Sanofi Genzyme, give them a lot of kudos, have really pushed this forward, and now they're in a phase 2 study looking at efficacy. Many more gene mutations. This is VPS35, and I'm not going to go into it. This is data that we've published recently. This is a Canadian family with receptor-mediated endocytosis 8 variants. Again, very interesting discoveries. This is another Canadian family, because I work in Canada. This is DNA JC12. It's a cause of early onset Parkinsonism, and it was families that came out of Italy and Saskatchewan. Again, I'm not going into it, but I'm going to leave you with this: that I believe that, you know, there's a saying: if it swims like a duck, it walks like a duck, and it quacks like a duck, it most probably is a duck. And And all of these rare families with Parkinson's disease, if they're manifesting with late onset, typical garden variety clinical Parkinson's disease, and many of them have the same pathology as garden variety Parkinson's disease, but nevertheless they have a genetic basis for their condition. Perhaps those genetic forms of these genetic clues are pointing us to the same common pathways and processes within cells, within neurons, that have gone awry. And it seems right now that many of these very big genes and very important players are basically pointing to endosomal trafficking. And there's some two aspects. So endosomes are these membrane compartments. You can see a picture up here on the right-hand side that are basically be responsible for shuttling cargoes, protein cargoes, within the cell from one part of the cell to another. They're involved in synapses, which is what's shown here, in terms of recycling proteins back to the surface rather than degrading them. And they're involved also in lysosomes, in basically shuttling proteins that are destined for degradation, for breakdown into lysosomes. That's shown on this schema. And all of the genes for Parkinson's disease, pretty much, many of them, typical garden variety Parkinson's, late onset Parkinson's disease, basically point to a breakdown in different parts of this pathway in this system. So I think it's leading us to a, if you like, a grand unified understanding of what Parkinson's disease is from a molecular perspective. And that's giving me a lot of hope and encouragement, and I hope you too, that we will be able to fix this condition and ultimately prevent this disease, if not halt its course in people who are already affected by it. I'm going to open up to questions now because we've got about 10 minutes left, I believe.
Alrighty, wow, that was a very informative presentation. I have to admit, I think genetics has come such a long way, and it's just really exciting to see the possibilities of where it can go that, you know, will help people with Parkinson's. So thank you so much, Dr. Fair, for sharing all of your knowledge and your expertise. I think it brings peace of mind to a lot of people. So at this time, if anyone has any questions for Dr. Fair, I do suggest if you look at your bottom panel, you'll see a question area that you can ask your questions.